So first, I'd like to introduce Associate Professor Erin Barreto. I would like to say that from the outset that I was fortunate enough to dial into her Viva for her PhD a couple of years ago. I can't remember what time of the night it was here in Australia, but she kindly invited me to listen in, and it was a fantastic description of a brilliant body of work, which was not only complementary but which intertwined and interconnected with each other very well, all in and around this topic about what she's going to be talking about, which is consensus recommendations for use of individualized drug dosing in patients receiving beta-lactam antibiotics. She's very well published. She has very good professional and clinical recognition for her skills as a clinician scientist as well, and has excellent grant funding as well. So it is my pleasure to welcome Erin to present.
Thank you so much for the chance to be here. And it is night here in Minnesota, and so I'm pleased that my children are down and I have plenty of time to talk to all of you. So thank you so much. I'm grateful to be sharing some of this work from the IBLICS consensus group. I'm one of many people representing that group, and so I'm just happy to speak on behalf of the team. So we are going to cover the need for beta-lactam optimization, and then we'll talk about the recommendations from the group. As is probably very well known to this group, beta-lactam optimization is important because we know that from observational data that there's tremendous inter-individual variability in beta-lactam concentrations that are observed in acutely ill patients. Now, when we look across the literature, there's more than 400 PK models that have been developed for more than 40 different antibiotics, and they show huge variability in volume distribution and clearance in the population. And when you look even at the individual level, it's even more substantial. So these are now data that are more than 10 years old, well known to this group as they came out of this team primarily from the DALI study, which essentially shows that in routine clinical drug dosing with beta-lactams, the trough concentrations observed as shown on the y-axis here in the logarithmic scale are highly variable in patients with routine clinical doses. So you can see if you just look at the piperacillin tazobactam that concentrations of 1 to all the way up to 200 milligrams per liter are observed. And even if you just narrow that down to the bulk of patients in the interquartile range, you're still seeing 10-fold variation in concentrations observed. So for that reason, we undertook the development of the Beta-Lactam Individualization Consensus Statement. And before we jump into the overall experience of doing that, it's important to define what beta-lactam dose individualization is. So the consensus group determined that dose individualization reflects a dose regimen for a patient that is informed by measured concentrations from that patient. Now, we all know that we've done dose individualization for many years based on age and weight and creatinine clearance. But fundamentally, these are kind of population-level interventions because we know that still, even with GFR-adjusted drug dosing, we see substantial inter-individual variability in concentrations observed. Therefore, we believe that for this particular concept of beta-lactam dose individualization, you really do need measured concentrations from the patient. And there's lots of ways that you can accomplish that through model-informed personalized dosing nomograms, decision trees, and then, of course, therapeutic drug monitoring. The goal of this consensus statement was to kind of undertake an effort to summarize contemporary evidence that looks at the outcomes associated with beta-lactam dose individualization and then key implementation and technical considerations that must be accounted for. And the target audience was healthcare professionals caring for patients treated with beta-lactams, focusing on hospitalized patients, and really anybody across the age spectrum from neonates all the way to the older adult. We also took any systemic beta-lactam antibiotic, enteral or parenteral, and treated it the same regardless of infusion rate. So we understand, of course, that prolonged infusions and loading doses and other strategies are tactics to improve beta-lactam individualized dosing. So we think that these recommendations fall alongside and in partnership with the existing recommendations for prolonged infusions that are out there. They certainly do not conflict. Now, this is the first time actually that we're sharing these recommendations publicly. They are approved by 6 endorsing organizations, and we're waiting for the final determination of the 7th before we can move forward with publication, but we feel good about the statements that we've made. These groups represent the international community in critical care, infectious diseases, personalized medicine, and pharmacotherapy. The IBLICS panel was a really diverse panel. We're very proud of that. I included 19 subject matter experts with the disciplines that I just mentioned, as well as laboratory medicine with representation from clinical chemistry, clinical microbiology, pharmacometrics, and people with guideline methodology. We also included 3 consultants for the project, a librarian, a biostatistician, and then a patient partner to make sure that we keep focused on really the priority for why we're doing this work. And then we had a really nice representation. This is a global effort, as you can see on the map, with people from all over the world. We had different levels of expertise, all ranges of race, ethnicity, and then different genders. And the goal of the group was to really focus on 3 key themes: the clinical impact of beta-lactam dose individualization, the key requirements for beta-lactam dose individualization, and some of the technical and methodologic considerations that we need to think about. Where possible, when the evidence was able to support it, we used grade level recommendations, where we used the typical language of the panel recommends for a strong recommendation, the panel suggests for a weak or conditional recommendation. And in this space, there is really a lot of heterogeneity in the evidence, and so there was not that many recommendations that were gradable. And so in those circumstances, we proceeded with issuing best practice statements where we could. Now, there was a lot of questions and a lot of recommendations and statements made in this guideline, and I can't cover all of them, of course, today, but I'll talk about some that I think are probably of greatest interest to the group. The first of which relates to the role for beta-lactam dose individualization on patient-centered treatment outcomes. So this effectively is the efficacy question or effectiveness question. And after review of the evidence, the panel issued a conditional or weak recommendation to suggest the use of beta-lactam dose individualization to improve patient-centered treatment outcomes, specifically motivated by the outcomes with clinical cure and mortality. As you can see, at least for clinical cure, it's quite convincing that there is an association between beta-lactam dose individualization and cure. With mortality, it does look to be a signal in favor of benefit, and the probability of benefit, even with skeptical priors, looks quite good. But the recommendation was downgraded because there was a few randomized controlled trials, really only 4 out of the 11 studies that formed the basis for this recommendation. And although there was a consistent signal of benefit, particularly with the cure endpoint, some of the other endpoints, it wasn't quite as obvious, including length of stay or microbiologic cure. There was not any evidence of harm, and so ultimately that's what motivated the lower level of evidence quality and or evidence certainty, excuse me, and the lower strength of the recommendation. And the group certainly acknowledges in the rationale for this particular question that while this is a recommendation in favor of beta-lactam dose individualization, it needs to acknowledge and recognize the constraints of resources that some places have in actually implementing this. Another key question related to PK/PD targets. This is a popular one. What target are you going to choose for your beta-lactam dose individualization program? And the panel suggested that a minimum PK/PD target of 100% free time above MIC during the dosing interval was recommended to improve the likelihood of clinical cure. One of the things that was considered in here is that there was a lot of heterogeneity in how MICs were applied in studies. Some were assumed, some were measured, and the measurements occurred using various laboratory platforms or were not even reported. Now, certainly some people like to discuss the potential of using 100% free time above 4 times the MIC, but the panel felt that this would likely lead to very high doses used and certainly would need to be tailored to a specific illness severity or infection type or type of organism to make a determination rather than widespread use of that for all patients. And so the recommendation here is to use 100% free time above MIC as the minimum threshold. So that's the effectiveness side. On the safety side, the panel was not able to make a recommendation on the role for beta-lactam dose individualization to improve safety outcomes, but did in fact recommend a best practice statement that favors monitoring for adverse events, particularly neurotoxicity. I think some of us were a little bit surprised at first when looking at this absence of a recommendation for beta-lactam dose individualization to improve safety. But I think if you step back, it's not altogether unexpected. These events are quite rare. Neurotoxicity occurring in 2 to 5% of patients, nephrotoxicity and hepatotoxicity being more uncommon and really not likely dose related. And so it's very hard in an unselected patient population, a standard dosing trial, to be able to detect some major safety signal. And so we just were not able to issue a recommendation on that. But there are certainly key patient considerations that you would want to notice. And in the consensus recommendation, we do highlight that patients with kidney dysfunction, patients with patients who are elderly, patients with prolonged courses of treatment, all would be at higher risk for adverse events and deserve additional monitoring. Another question is what patients to do this in. Now, there's not an RCT that says you should or should not do this in these exact patient populations, but when you look across the totality of the evidence here, what you start to see is that really there are two big groups where this has the most evidence of benefit, and that's in the critically ill population, particularly those who are on some sort of extracorporeal support device, and in patients who have something going on with their kidney health, whether it be acute kidney injury and chronic kidney disease or alternatively augmented renal clearance. And so the consensus statement favors the use of beta-lactam dose individualization specifically in those two groups. There are obviously many other groups that have reports of use of beta-lactam dose individualization, and broadly it's individuals who have PK/PD abnormalities or very difficult to treat infections, some of which you can see highlighted here as specific examples where there are data for the use of beta-lactam dose individualization. And so the panel suggests that people consider it in those circumstances. Now, I don't think any of us are naive that this is an easy proposition. Dose individualization, particularly if you don't have a program already built, is challenging. And I think this is one of the major sticking points with a recommendation in favor of this, with the professional organizations endorsing it. And so we made a very concerted effort to highlight the challenges and strategies to implement this into clinical practice, focusing on who you need to have at the table, as well as some of the key considerations and integrating it for the clinical workflow. And implementation of this is a whole talk in and of itself, so I'll spare you the details. But suffice it to say that the consensus recommendations, once released, will provide a framework that can be adapted, as well as some key references that people can reach out to try and understand how they could bring such a program like this to their hospital in light of these recommendations. The last recommendation that I did want to highlight during this presentation relates to the pathogen MIC. So this is quite a hot topic, particularly as you try and interpret the results of different studies where MIC was applied variably. The panel supported the use of a laboratory-defined MIC, but if the reported MIC is within the susceptible range, the panel recommended the use of the susceptibility breakpoint of the ECOG. Now, this is to acknowledge the fact that an MIC is really just a single number that represents the population of bacteria, and there's inherent variability in that number. And so, several doubling dilutions below and above can be kind of the range of susceptibility within the population. And so rather than chasing very low numbers of very highly susceptible organisms on the surface level, rather we advocate for just treating to that kind of susceptibility breakpoint or the ECOG. So the example here would be if you had a patient who has a Pseudomonas aeruginosa bloodstream infection with an MIC of 0.25 to mirum, if the target, in accordance with the consensus recommendations, is a free time above MIC of 100% of the dosing interval, then theoretically you would be targeting a level of 0.25 or higher at a trough. But in reality, the true population of bacteria that you're looking at has some sort of range in their MIC values. And so rather than chasing all the way down to that 0.25, we favor the use of a free marrow level of at least 2 to account for some of that variability. And of course, also to account for things like penetration to the site of infection. This is just a small snapshot of the 16 recommendations and best practice statements in the IBLICS Consensus Statement. Like I said, it's going to be released sometime in the next month or 2 months or 3 months. But, I think broadly what it argues, I think convincingly, is that there are certainly issues with ongoing variability across patients and within patients in their beta-lactam concentrations, and that variability translates into clinical outcomes that can be improved with the use of beta-lactam dose individualization. And so sticking to that and adhering to some of the key considerations when it comes to implementation in the laboratory principles can help hopefully move us a step closer to personalized pharmacotherapy.
Thanks so much, Erin. That was an excellent presentation. Very topical. I'm sure that everyone who is listening in would have really enjoyed that. Provides some really good information about what the consensus is on this topic, and we look forward to that being shared a bit more broadly in due course.